Good morning or afternoon, class. I wanted to move forward in the notes. Hopefully, you have done the assignment for dihybrid crosses and monohybrid crosses following basic Mendelian genetics. Today, I wanted to go forward into the notes into the exceptions of the normal rules for Mendelian genetics, the irregular stuff, the things that don't follow the normal rules. If you could, pause the video and pull up a new window with the notes. You'll find it in the Google Classroom under the classwork at the top. On the left, you'll find the different units. You'll find heredity. And at the bottom of all your choices for the file for heredity, you will find the PowerPoint. I am on slide 39 that has the title says Mendelian Genetics Lab Survey. So normally, I would survey y'all as a class and see which traits you have. On that slide, there's some pictures. There is the attached and detached earlobe. That is a Mendelian trait. Some people have an attached lobe, and some people have a detached lobe. A detached lobe means that your lobe hangs down further, and there's a gap between the bottom of your lobe and the side of your skull. There's also the cleft chin that creates an indentation on the end of your chin. There is the widow's peak that you can see in the bottom left with a guy holding the giant needle. That's Jake from Vsauce 3. And he has a widow's peak. I don't have one, but a widow's peak would be a dip down of your hairline along your forehead. And then also, of course, a fun one is whether or not you can curl your tongue. That is another Mendelian genetic trait. So the next slide after that is variations. It has a picture there for paternal and maternal chromosomes, so they're color-coded. Paternal being there in orange, and then maternal being in garnet. And you can see the process of crossing over, where while they form a tetrad, where the two different chromosomes get bound together into one mass, called a tetrad, sometimes pieces of the genetic code will break, and the cell will freak out, and enzymes will reattach them, sometimes in the right place, but sometimes in the wrong place. And if it is in the wrong place, we call that crossing over. Some species are more likely to have crossing over than others. In some of our plants, especially corn, crossing over occurs much more often than in the human species. But we still do have it happen occasionally in our genetics. So the next slide is a definition for crossing over. It's a process by which alleles are exchanged from one chromosome to another. Moving forward, by the way, the crossing over creates variations, changes our probability of getting certain traits when we do our Punnett squares. The next slide is two different terms that my students in the past always get confused. So I'm going to try to explain this the very best I can. But they are very similar sounding, but they create two different products. There's incomplete dominance and codominance, and I'm on slide 42. Incomplete dominance and codominance are not following the normal Mendelian genetic rules. So let me start with incomplete dominance first. Right now it is spring. You can go to maybe your local park if they're still open. You can go to maybe Swan Lake. And some of the flowers on the bushes right now are azaleas. Azaleas are very common down here in the south. And they're beautiful and come in different colors. There's white, there's pink, there is some reds and even purple shades of azaleas. And they're a great example of incomplete dominance. An incomplete dominance, if you take, let's say, a red azalea and cross it with a white azalea, you will not get a red or a white offspring. Instead, you get a pink, somewhere in between. So it is incomplete. It's not completely red and it's not completely white. It is somewhere in the mix. Incomplete dominance. I'll show you how to do that on a Punnett square in just a little bit, but let me go on to codominance, so I can show you both of those at the same time on a Punnett square. Codominance, a good example of that is camellias. And camellias are in a lot of people's gardens. There is even a section on the southern side of Swan Lake that is called Camellia Island. It is a man-made island, and it's covered with camellia bushes, and there are several on the north side of the gardens as well. Camellias typically bloom in December, but there may be a few still blooming right now. And they are a great example of codominance. Codominance is where you take, let's say, a red flower and a white flower, and you cross-pollinate them, and the offspring will show both red and white. Like you see in that picture there is a beautiful flower with red and white in the bloom. So both characteristics are being expressed. Codominance, both are there. So let's do that on the Punnett square. I have incomplete dominance and codominance. So we can't follow the normal rules for our shorthand for our genotype. Instead, I've got to use a different form. I'm going to use I, but you could use other letters as long as you can differentiate between the capital letters and the lowercase letters. So I can easily make the difference between a capital and lowercase in our incomplete dominance flower here. Now, there's two dominant traits. I'm sorry. Let me fix that. There we go. There's two dominant traits, both of them with capital I's, and I have a W for white, and I have a R for red. And if we were to do the cross and plug these in, I'm going to end up with by the way, with the incomplete dominance, the order that you put them in doesn't really matter as much. If there was a recessive one, the recessive ones come out second. But in this case, there's not a recessive one, so we can put either first. But it's good to be consistent. If you put R's first, stay with R's. So I have capital I and an R, and a capital I and an R, so this will be a red flower that got the red trait from dad and the red trait from mom. I have at the other end, I have capital I with a W and capital I with a W. That's going to get the white trait from dad and mom. And this is going to be a white flower. But both dad and mom I made as pink. So it's not really red or white. And with crossing a pink flower with a pink flower, I could end up with 25% red, 25% white, or 50% at the other two ends with red and white, so that will make pink. These two are going to be my pink flowers. Now for codominance. And I need to fix this too. Codominance will look exactly the same on the Punnett square. That's why you've got to just know which instance you're dealing with. Are you dealing with codominance or incomplete dominance to figure out the phenotype? Finding out the genotype with a Punnett square is easy, but when we're dealing with some exceptions to the rule, we need to know what instance it is. Is it incomplete or codominance? So these two are identical. For incomplete dominance, I have a flower that is red and white, that's the father, and a flower that's red and white, that's the mother, and I cross them. If I cross them, they can't pass on both traits, so I could end up with a flower that is completely red, one that's completely white, so 25% for red, 25% for white, and then 50% of them is going to be red and white. Comes out exactly the same, but they're going to look very different. We had a 50% chance for a pink with incomplete dominance, and a 50% chance of an offspring that is red and white with codominance. And I'm using these capital letters to help indicate that it is a dominant trait. If I was to use lowercase i's, that would mean it would be a recessive trait. But with incomplete dominance and codominance, they don't have a recessive trait unless it is a special case. The next slide says why women are stripy. I'll attach that to this file so you can watch that. And the next one after that is multiple alleles. So you may be aware that there is different blood types. There is A, there's B, there's AB, and then there's O. So those are the 4 different blood types, AB, and O. And this is different alleles. You can get the type A, you can get type B, or you can get type AB. If you get type AB, it's not just A, it's not just B, it's not somewhere in between, it's both. So that would be not incomplete dominance, that's codominance. Both are being expressed, A and B. And then we get O, which is the odd one, I'll come back to that. So, how does blood type work on a Punnett square? Well, A is a dominant trait, and B is also a dominant trait. So let me show you what this looks like on a Punnett square. Let's say that Dad is the blood type A, but Mom is blood type AB. She has A and B. If we were to cross that with codominance, we're going to end up with 50% of the children being blood type A. My top 2 are type A, because they got A from Mom and A from Dad, so they're going to be type A. But at the bottom, we have a 50% chance of the offspring being AB, just like Mom, getting the A type from Dad, and then getting B type from Mom. So 50-50, either going to be type A or AB, and this again is codominance, because both are being expressed, A and B. Now, what about O? O type is different. O type is actually recessive. A is dominant, B is dominant, AB is dominant with both of them, and then there's type O. So let me plug that in to show you what that would look like on a Punnett square. Since it is recessive, I can't use a capital I, because capitals are to indicate a dominant trait. Instead, I'm going to use a lowercase I. Let's say that Dad is type A blood type, and Mom is type B blood type, but they're both carriers for O. So Dad is A with a carrier of O that's not being expressed because it's recessive. Mom is type B, but a carrier of O, not being expressed because it's recessive. What do we end up with for our children? Well, in the very first top left box, we have a 25% chance of getting a codominant AB blood type child. Then the next one would be a type B blood type with a carrier, or a chance, carrying the type O gene. The next possibility is a type A blood type, also a carrier for O. And then the last one would receive the recessive from both parents, lowercase I, which would be O. You might be wondering, why do I have an A and a B beside the 2 capital ones, but no O beside the lowercase? Because there's only one recessive blood type, and that is O. So anytime I do an I and I know I'm talking about blood type, I know it's O. But for the dominant ones, I gotta specify, because it could be A or B, so I need to let myself know. But in this cross, a father that's type A, a mother that's type B, we could end up with 4 possibilities: AB, B, A, or an O blood type. That is using a different variation than your typical Mendelian genetics and crosses on Punnett squares. So going back to the slides, I'm on slide 44 and just explained blood type a little bit. I do want to mention, you probably have heard of type blood types that are positive and negative. That's another antigen. On the outside of our blood cells, red blood cells, are little proteins. So these little proteins are like identification cards. Like you walk around school and you have your ID shown. Those little identification cards come in different shapes, and there's shape A for A blood type, shape B for B blood type, and then if you have both A and B, you're AB blood type, and then if there is no identification card protein on the outside, then you're O. It's missing it. This is why if you are type O blood type, you are the universal donor. But what about positives and negatives? Well, there is a second set of proteins that if you do have it, you are positive. If you do not have it, then you are negative. So you could be A positive or A negative. You could be B positive or B negative, or O positive, O negative. It doesn't matter. Those are independent of one another, so you'd have to check for both. And that would be on a separate Punnett square. Sorry that the camera keeps moving around, the cat keeps jumping on the table. Polygenetic traits. This is when it takes multiple genes together in concert to make a product, a phenotype. Like for example, I have here skin color. So your skin tone is not made by just one set of alleles, one gene. Instead, it's a couple dozen different genes that determine your skin type. So you can inherit from mom some traits that will make your skin type, and you can also inherit from dad some traits to make skin type, and that can create an interesting combination in what your skin will look like with the shades and hues. Next slide is just a picture of the types of blood, and you'll notice at the bottom that it has the donors. So if you're type O, you're considered a universal donor because it doesn't have those IDs on the outside of the cells, so you can donate to other O people. You can donate to AB or AB. But if you are a type O blood person, which I am, that means that you are the unlucky few that can only accept type O. Because if I was, for example, to do a blood transfusion and get someone that had type A or type B or type AB blood, then my body would recognize those IDs on the outside of the cell as something foreign, something that shouldn't be inside my body, and will start attacking it. And the blood will clot, and it could cause organ failure, and if it's too much blood, it could lead to death. This is why Red Cross and other facilities try to get people to donate type O blood, because they need it for other type O people and can use it for anyone else. Because it's like a ninja. It hides in the body of the other people. If you're type A, B, or AB, and you receive type O, it doesn't get recognized as a threat. It doesn't have an ID there on the protein outer coat to show that it is something that shouldn't be there. I have there on the next slide on 46. The example of the blood type, and you see that dad at the top is type A and mom on the left is type B, but is also a carrier for O, and you see that 25% of the offspring are going to be AB, and 50% are going to be AB, and 50% are going to be type A and carrier of O's. Alright, sex-linked traits. That's going to be a long video, sorry. So sex-linked traits are interesting traits that sit on our sex chromosomes. We have 23 pairs of chromosomes. 23 came from dad and 23 came from mom, and that's our pair. That last 23rd, one came from dad, which could have been an X if you're a girl, or a Y if you're a boy, help determine your gender. Mom donated an X chromosome. So if you're a boy, you got an X from mom and a Y from dad, XY. If you got, if you're a girl, then X from mom and an X from dad. Those last chromosomes is our sex chromosomes. They determine gender, they determine a few other things. And one is some of the shapes for the rods and cones in the back of our eyes. So if you were to inherit a irregular gene from one of our parents, then it may affect your ability to perceive colors. or be colorblind. Sex-linked traits are very interesting. For example, color blindness shows up much more in men than in women, and I'll show you why when we get there. The next slide says in humans, the Y chromosome carries 26 genes. That's it. Whereas the X chromosome contains slightly over 1,000 genes. So that's why males are dependent heavily on what's on that X chromosome that they got from mom. Because on the Y chromosome they got from dad, there's only 26 genes. That means that there's not a backup for a lot of the genes that you get from mom. Mom's got the one and only copy that she donated to you. If you were to have a backup, then it may mean that you wouldn't have certain traits like color blindness. Going forward through the notes, I have a colorblind test where you can see some things in the colorblind test dot pictures. And then I have a slide that has a family. On the far left in orange is Kyle. Next to him is Susan, the mother, and then Brooke with the skirt. And the last one in the blue polo is Cody. They're a great example of sex-linked traits. The father is not in the picture. I think he was one taking the picture. Ricky. Ricky is not colorblind, and I have him there on the Punnett square. He's male, so he has an X chromosome and a Y chromosome. The Y chromosome does not have the trait for making the rods and cones in the back of the eyes. So I have that as a capital C. So he's not colorblind, and the Y chromosome has no C with it because it doesn't carry that gene. Susan, in her family, color blindness is a issue for some of her brothers, but she is not colorblind. If you can look very carefully, she has XX, one with a capital C and one with a lowercase c, because we know she is a carrier of color blindness, although she is not colorblind herself. Now, her kids. The first one born was Cody, the one in blue. Cody is colorblind. He got a Y chromosome from his father, Ricky, made him male, and then he got, out of the 2 X chromosomes from his mother, he got the one that has a bad copy of the gene that led to color blindness. So he is colorblind, still drives, he works, he lives pretty much a normal life, but he has trouble distinguishing between some shades of greens and browns and a few other colors. But it doesn't mean that he has a seeing-eye dog. It doesn't mean that he can't function like a normal human. It's just a few colors he can't distinguish easily from. The next child was Kyle. Kyle is not colorblind. He got the Y chromosome from his father, and he got the good X chromosome from his mother that has the proper genetic code for making rods and cones in the back of the eye. Then the third child, Brooke. Brooke, we don't know what her genotype is. You see, she's not colorblind, but we knew she wouldn't be anyway because she got an X chromosome from her father, and her father wasn't colorblind. So he passed on a good X chromosome to her. But we don't know which X chromosome she got from her mother. She could have gotten the good X chromosome, so she has 2 good X chromosomes that don't have the trait for colorblindness, or she could be just like her mother and receive 1 bad X chromosome that has the trait for being colorblind. We don't know. But this picture that you see in the slides is fairly old. Everyone is much older now. They're my age. And Brooke has had children. Unfortunately, they're still too little for us to test to see if they are colorblind. But just because they are not just because they don't show signs of being colorblind doesn't mean that that she doesn't carry the trait. She has 2 copies of an X chromosome, a good and a bad one. It is luck of the draw whether or not she donates the good or the bad one to her offspring. She could be a carrier and just not have kids that get that bad chromosome. But if any of her kids do turn out to be colorblind, then we know for certain that she is a carrier because her husband, Alex, not in the picture, is not colorblind. I'll also link the ASAP Science video on hair loss balding, which is a sex-linked trait as well. You might be thinking color blindness shows up much more in males. Balding is much more a common trait for males. Why is it all males? Well, that's because females get 2 X chromosomes, one in the backup. So if there's one bad copy, they have a good one. And males only get 1 copy. So that's why a lot of these traits show up way more in males than it does in females. Alright, I believe this is where I'm going to stop this video for today, because it's already been 25 minutes, and I want to do one more video after this to close out the unit about genetics, keeping track and engineering, and some of more strange things. So thank you for following along on the slideshow, and I hope everything makes sense, and stay tuned for another video.